We are recording. So for everyone asking, hello, welcome to your first Friday tutorial. Happy to have you all here. One thing, actually, we'll do an introduction first. So my name is Chloe Mitchell. I'm a student in the Deeper Lab. I'm in my 5th year, oh my God, of a PhD. So my work is kind of, I guess, structural biochemistry, but also somewhat drug discovery. I'm doing antibiotic drug resistance, looking at kind of new ways to tackle resistant mechanisms in terms of coming up with new therapeutics towards that. Yeah, so that's a bit about what I do. I work at SickKids, that's what I've got my lanyard on. Yeah, so if anyone has any questions about grad school, I just want to kind of upfront say, please email me and let me know. I'm super happy to talk to you about it. Okay, course syllabus, everything's on Quarkus that you need to know. Everything's in the syllabus. Please read the syllabus. Tutorial notes are on Quarkus. So they're going to be posted at the beginning of each week, and only Fridays tutorials are going to be recorded, so just this guy. So all the students asking, yes, it's recorded, it will be posted, so if you do miss something, or if you can only stay for an hour, you can come back and see what you missed. For participation, so we're going to be doing polling options. So I have several kind of questions as we go through, and I'm just kind of doing one right now. So by participating in the poll, it's going to record that you were at lecture. So you just have to kind of click these polling options for at least like an hour. So I'm going to post these as often as I can so that you guys can get those participation marks. Absolutely. Course. And okay, so we're going to try one. Speaking of, okay, start new. Okay, we're going to try this. Getting you participation marks. Are you guys excited for the bachelor? Who? No? Oh, I'm so upset. How do I move this off my screen now? Okay, so I guess, oh, like the show The Bachelor, not like a bachelor's degree. Okay, so you guys are now getting the, like, so this is going to start tracking your participation, and then as long as you kind of respond to these polls within an hour of some point of this tutorial, you will get your 1%. Love Island, I do agree, Love Island is trash, but I do kind of love it. Okay, 23 of you haven't responded, so I guess you'll get the participation marks on the next one. Okay, great. You guys are doing well. Too hot to handle. Love our trash TV. Okay, please use the discussion board as you guys are going through. I know a lot of people have a lot of questions, so everyone's probably gonna have the same questions. So please post them on the discussion board. However, you can also feel free to email me. Okay, breakdown of tutorials. I'm gonna try and do about a quarter of it review and the most of it practice problems. If you guys want a shift in this, or you want more review or anything like that, please let me know. This is for you guys, so want to tailor it to you. I'm going to try my best to leave some questions for Q&A. I think some of the other TAs went a little bit over, so we're going to have to judge that accordingly, but we're going to try and have at least like 10 minutes at the end for students to ask questions. And if you have any ideas to help me make this more accessible or like easier for you guys to do, please email me and let me know. Okay, we're still talking about reality TV. Okay. How do we submit questions to the poll? Just by answering them, Quarkus Real or BBB Collaborate is going to tally it. Did you share your email yet? I think my email should be in the tutorial section, or if you like click on my name, I have like my contact info. So if you guys, yeah, it's also in the syllabus. Thank you. Okay. Okay. Strongly encourage participation, which I guess I don't have to tell you guys because you're quite chatty. This is lovely. So please feel free to respond in the chat and ask questions. Again, questions on the discussion board, email me. And one thing I do want to try and do is, because I know this course, there's a lot of material and it goes a bit fast. So if we did cover something, let's say this week, and you've been through all the lectures, the quizzes, my tutorial, and it still isn't clicking, please feel free to send me an email, and I'm going to try, I can try and review it in the next tutorial. Go over any topics that I might be getting a lot of emails about or I'm seeing on the discussion board a bit. So if there's something you want me to revisit, I'm happy to do that because I think it's important because this course really builds, especially in the first unit. So if there's something you're not understanding in 1 week, it's probably going to pop up again. Okay. Tips for success: watch lectures early. Please try all the questions. Come to tutorials. I like hanging out with you guys. Look on the discussion board. Also, there's a lot of posts on the discussion board, so do please try and read ahead and see if any student has already asked your question. Also, I love the participation on the discussion board right now of you guys answering questions. I think that's great. So I love that you're answering each other. Love that. And yeah, start your assignments early. I think someone asked a question. Do we know when is a good time to ask? For assignments, yeah, direct those to Dr. Patterson, just because she's the one who kind of made the assignments, and I don't want to give you any false information. If you have questions about content in the assignment, you can feel free to ask me. But if it's like kind of details about the assignment or kind of the little nitty-gritties, direct that to Dr. Patterson. Okay. Brain break questions broke my brain. Mood. Oh my god. Okay. Solutions to the practice problems aren't going to be posted. We're going to be going over some in tutorial. If they're not covered, please post them on the discussion board and we can all go over them together. Okay. Feedback on the assignments, probably not, but you guys have your peers and you can feel free to, you know, I guess, or maybe that's cheating. Okay, maybe I don't know. You you can probably talk to each other and ask for help. You're all going to be having different proteins, I guess, at the beginning. Okay. Problems we're talking about. Okay. There's a problem set of practice problems that I'm going to be going over some, and then there's some extra in the tutorial. When are we getting the assigned proteins? I'm not sure. You'd have to ask Dr. Patterson about that. Okay. I'm going to try to get into some content. Okay, it's okay to get feedback. Okay, good. Yeah. I can't unfortunately give you guys feedback though on your assignments. Okay. Takeaway points for today. Okay, is there any other, I guess, logistical questions? I'm trying to. Yes, please don't copy each other. Thank you. Okay, sweet. If you have any more logistical questions, hopefully we can get to those at the end. Okay, takeaway points for today is fundamental interactions, bit of chemistry review. I think you all are supposed to have taken chemistry, so highly recommend you do a bit of review on that because that's going to come up all throughout protein structure and interactions because that's what's obviously driving this. Speaking of driving forces, so our molecular interactions, our hydrophobic effect, ionic, H-bonds, local environment, pKa's, all that stuff. It's good to have a really solid understanding of this. Going forward, it's really going to help you. And then the amino acids, I know there's 20, but you got to know them all. They're going to pop up throughout this entire course. So if you can hear an amino acid, see that like 3-letter code, 1-letter code, and like know exactly what the properties are, it's going to really help you a lot with your success in this course. So I really, if I can like get anything into your brains, that's what it would be. Okay. Quizlets. Yeah. Okay. All right. Functional groups. So we have our hydroxyls, carbonyls, our carboxyls, amines, thiols, or sulfonyl hydrides. So these groups are going to be really important. So the fact that they can be ionized, so that just means they can be charged. So we have oxyl group and our amine group, as well as our thiol can be deprotonated. Our nucleophiles and electrophiles are electron acceptors. If you guys remember back to your chemistry days, remember we have this carbonyl, which is great for undergoing nucleophilic attack, which we're going to see. So let's say we took this hydroxyl and it has its lone pair. Our oxygen has a partial negative, and then this is going to kind of be electron withdrawing. So we're going to get a partial positive on that carbon. So therefore, this lone pair, that electron negative, is going to want to attack that carbonyl. So we're going to see this pop up a fair bit. And then hydrogen bonding acceptors and donors. This is really important in stabilizing protein structure. Do we need to review chem? I would review a bit of just your hydrogen bonding and some of your nucleophiles and stuff like that. Okay, hydrogen bond. This is going to be occurring between what types of groups? So we have acceptors and donors. I'm expecting the chat to like totally blow up, but what do you guys think? Yes, it's going to be recorded later. So what qualifies, let's say, a hydrogen bond acceptor? What do you need? Electronegative atom, lone pair, love it. Yes. Boom. Okay, what about donor? Electronegative bound to an H bond. Yes. Okay. So we're going to have hydrogen bond donors. So we need that electronegative atom. Again, it's pulling that kind of electron density, so you're getting that kind of induced dipole or that partial positive charge on that hydrogen. And then that's going to be attracted to that partial negative charge through the lone pair of that electronegative atom. Now, I think I saw some students talking about this on the discussion board. There's going to be better electron donors and acceptors. Some can form them, but they might be a little bit weaker. So there is, not everyone is going to be equally as strong. Okay. Hydrogen bond donor acid. Yeah, so I guess the carboxyl group, technically it could be a donor, even though it's probably most likely going to be deprotonated, but yeah, along those lines, depending on the state, the protonation state that it's in. Okay. What kind of non-covalent interactions can an ester form? H bonds, yes. Someone said van der Waals. Yeah, I guess we could participate in packing. A lot of H bonds. Yeah, so we're gonna have our H bonds. So again, you should know what an ester looks like. And we're gonna have our 2 lone pairs on our carbonyl, as well as our 2 lone pairs on our oxygen here bound to our R group. So these are gonna be H bond acceptors. So I think the carbonyl is the most predominant when esters are found, and this group is not so much for being the H bond acceptors, but it definitely can form hydrogen bonds. Okay, I want you guys to be able to recognize functional groups. So looking at guanine, how many functional groups can you guys identify? I'm gonna pull this. Get you guys your participation marks. I have to keep exiting this out each time. Okay, whoa. Amine. What type of amine? We've got primary, we got secondary, we got tertiary. Okay, someone says secondary. Someone's saying ketone, but is there anything else that's unique about that ketone that might be beside it, making it something different? Amide, someone said it. Nice, yeah. Okay, amine, secondary, amine, and 9. Okay, yeah, you guys are, did I post? Did the poll get posted? Ah. Choice, update. No. Okay, why isn't this poll working? View poll. Oh, there we go. Can you guys see it? Okay, while you guys are answering your poll, I don't use. Okay, I'm gonna draw on this. So, oh my god, how do I? Responses for how many functional groups do you see? Yeah. So I'll leave this up for a second, so everyone could. Everyone gets their participation in. Poll isn't working. Imine. Yeah, someone said imine, that's good. Sorry, I don't know how to move this poll, which is gonna be interesting. Okay. Poll seems to be slowing down, so I'm gonna take this down because I can't draw on it, unfortunately. Oh wait. Oh, there we go. Okay. Bam. Sorry guys. Working through this. All right, so we've got a amine there. So this is going to be our primary amine. We've got an amide. Now I know this isn't a ring, so it's a little bit trickier. We got, someone said imine, which we got here, and then we got this secondary amine there. So remember, our amines are going to be primary, secondary, or tertiary, depending on how many of our groups. Poll results. No, there's no tertiary. That's good. Are you talking about the carbonyl here? Because the carbonyl on this 6 is beside the nitrogen, the amide, it's going to be an amine. It's a amide. Again, I know it's a little bit trickier because this is a ring. So yeah, there's definitely, you can point out just the carbonyl, but sometimes when they're together, you're gonna get like that's gonna, it could be a different type of functional group. Okay, this guy is called an imine. Imine. I think sister wrote I-M-I-N-E. Alrighty. Okay. Here we go. Stop that. So yeah, the correct answer was 4. Guanine means a base. Nucleic acid. All right, next question. Examples of hydrogen bonds. Which one can form, or which groups can form the hydrogen bond? There we go. Guys, there's a poll. But I'm really happy that you all said F. Yes. Lovely. Good work, guys. Okay. Yeah, so all of these groups are capable of forming hydrogen bonds. So this is an example of being able to see the functional group name, recognize the structure, and what properties that structure has. F. Great. Okay. Why is water polar but uncharged? Geometry. Love it. Short and sweet. Symmetrical dipoles. Great. Yeah, basically, someone said geometry. So we're going to be getting partial positive, yes, dipoles. You guys are on the ball. So we're going to get that partial negative charge on our oxygen, where we're going to get the partial positives on the hydrogens due to that bent geometry. So therefore, we're going to get a dipole. You do kind of get sometimes, you can get hydronium ions, but then that's, I guess, not technically water. So I guess if you wanted to be super particular, you could get it charged. But that's if you were to add another hydrogen. But mostly it's this induced dipole that we're going to see becoming very important in terms of protein structure and stability going forward. How do these this dipole help stabilize polar amino acids and protein structure? Okay, functional group ionization. This is something that I think is super important throughout this course, especially when we get into pKa's, which we are. So it's going to help you kind of understand why groups are charged when they're charged in certain environments. So if we were to look at our carboxylic acid, this is our protonated form, and then we can have it in the deprotonated form, depending on, come on to here we go, depending on the environment that it's in. So what we want to measure is our strength or our acid dissociation. So how strong of an acid do we have? So remember, an acid, if we go back to our chemistry, it's going to want to donate its protons. So if we have an acid, it's going to, if it's a stronger acid, it's going to take a lower pH for it to donate that proton. And we can measure this by looking at kind of at equilibrium, the population of species that are present. So we have, if we have more of our deprotonated form, we're going to have a much higher Ka. Now, for some reason, biochemistry, they love taking the negative log of everything. I don't fully understand the math behind this, but if we take the negative log of that, we get our pKa. So we're measuring, again, the strength of the acid. So if it is a strong acid, it's going to donate its proton. We're going to have a large Ka, and that means we're going to have a very low pK. Oh, someone's asking me to go back for a second. If you guys want to. This is recorded though, so you guys can go back later. Sorry, we do have a lot of content to get over. So again, remember, strong acid wants to donate its proton, large Ka, low pKa. So our pKa is a measure of acid dissociation, or how likely it is to give up a proton. So when we eventually get to our amino acids, the lower the pKa, the more likely it's going to be deprotonated. The higher the pKa, it's more likely to be protonated, because you're going to need a more basic solution to kind of like pull that off. So always think about this in terms of that proton wanting to be given up, or like how strong of an acid these are. Okay, sorry, I'm just. Okay. Downstairs, are you guys still talking about water? Okay, yeah, I think you're still talking about water. Someone said you can repeat that. The water or that? Yeah, so the lower the pKa, stronger the acid, the more likely it's going to be deprotonated. We're going to be going over some examples. So I put the derivation just because some students are interested in this. You don't have to memorize this or know this. It's just kind of for your information. So when we have an acid, it's going to depend not only on the pK of that functional group, so how likely it is to donate that proton, but also the environment that it's in. So if we're in a very basic environment, it's probably going to be found deprotonated with a strong acid. But if you have a really low pH, and let's say like pH 1, you're going to have a ton of protons around. Even though it's a strong acid, that group might still be protonated. So even it heavily is going to depend on the environment that we're in, so we can actually compare those two values of pH and pKa. And this is the Henderson-Hasselbalch equation. So this is, again, the derivation of how these two groups are related, but we can, so pH is going to equal pKa plus the log of the two species. So I just kind of, again, have a little box for you guys to remember. You should know these equations. And pK, acid strength, pH is a measure of acidity. Ooh, okay. In the chat. If If it has a high pKa, it means more basic, right? So we would need more basic solution to pull off the proton. Yes. So the higher pKa, the more it's a base. So it's going to want to actually like hold on to those protons. Higher pKa, more likely to get protonated. Yes. You guys are doing great. Share my screen. Can you not see my screen? Okay. All right. So this equation, if we want to know kind of exact concentrations of at a given pH, what is the percent or what is the concentration of our protonated versus deprotonated species, but we could just kind of generalize this a little bit if we kind of want to see, is this protonated or deprotonated? So if we take a look at the relationship, let's say we have a very acidic environment, so very low pH. So the pH is going to be lower than the pKa. What protonated state are we going to actually find this functional group in? Is it going to be protonated or deprotonated? Protonated, perfect. So the group's going to be protonated. So if you're below the pKa, it means you're going to have more protons floating around. So you're going to have a protonated species. What about if our pH equals our pKa? 50-50, yes. So it's going to kind of be in this like middle ground where it's like half protonated, half deprotonated. And lastly, for pHs above the pKa, deprotonated, lovely. So again, if you guys like to think about it, we have a very basic solution, and whatever that pKa is, it's more likely to kind of try and pull off that proton. So it's going to give up its protons. Perfect. You guys are doing great. Okay, we're going to use our Hendelson-Hansbach equation. This is a review question 12, an intense exercise, which I need to do more of. The pH of the blood can drop to 7.1, and the concentration of the, oh my god, HCO3 concentration is 8 millimolar, and the pKa is going to be 6.4. What is the concentration of CO2 in the blood? First of all, you have to identify which is your protonated species and your deprotonated species in order to correctly put that into the the equation. So is there 8 millimolar going to be the protonated or deprotonated? Deprotonated, yes. Okay, lovely. Okay, so our HA is going to be the bicarbonate, and then we're going to have our deprotonated version, which is going to be our 8 millimolar. And then we could plug this in to our equation. Some extra oxygen. Someone's saying, do we need to convert to moles? As long as you keep it in the same kind of range. So like if you show your answer in millimoles, you should be fine. But yeah, you can calculate it. Yeah, I think I'm seeing people say 1.6. Perfect. That's the correct answer. So if we plug that in, our pH, we were told 7.1, our pK 6.4. Put in our concentration, you're going to get 1.6. So how does this tell us about the carbon dioxide? What is bicarbonate? How do you get bicarbonate in the body? You're working out, your body's forming lots of carbon dioxide, yes, water and CO2. So that CO2 is going to get added to 2 water molecules, form into bicarbonate. Yes, so then this is going to be in equilibrium. So that's why you can see if this breaks down, that's where you get your carbon dioxide. It's in a 1-to-1 molar ratio, therefore the two are equivalent. Okay, the poll, the correct option. Okay, while you guys are reading this, I'm going to make the poll. Stop. There we go. here is the poll. Okay, so most of you guys are answering D, which is correct. A couple of you are saying A. That was kind of like a tricky one because it's kind of like they're actually negative logs. Sorry. But yes, the correct is D. B. Sorry. I think they're not making the quizzes. Why is B wrong? So for, I guess it's not, the measure of pKa isn't necessarily, it's not like a hard, this is an acid, this is the base, like we see in pH. It's kind of more of like a spectrum. Yes, so some people are writing in the chat here the how you get the formation of bicarbonate to carbon dioxide. Okay, just to kind of show you guys why this is true. We told you it's 50-50, but I want to mathematically show you. If we set pH and pKa to x, these two are going to cancel out. We get 0. If we take the inverse of log 10, we're going to get 1. Therefore, HA and A will be equivalent. So mathematically speaking, these are the same. So it's they exist in 1-to-1 ratio when pK equals pH. Okay. Oh, nope, we're going back. Okay. So that was a bit of a review of the functional groups. Okay, you guys are doing great in the chat. Sorry, I'm not responding to all these, but I love that you guys are responding to each other. If you do still have questions about some of that, maybe save for the end or send me an email, so I'll make sure I get through everything. Okay, so it's really good you have an understanding of ionization, these weak molecular interactions, because they're going to have a huge influence on structure. So we're going to go over our electrostatic interactions, which, hydrogen bonds, hydrophobicity, van der Waals. These are all going to contribute to how proteins actually fold, which is really cool because there's a ton of research that's going on to, how can we take that primary strand of amino acids and based on their properties, like predict what the final fold's going to be. So there's a huge field on this, and some people have been successful in kind of forming tertiary structures, but really cool stuff. So there's still a lot we have to learn, but there's a lot we kind of do understand, I guess. Okay, van der Waals. These are probably the weakest of the forces because you need the molecules to come really, really close together. We're literally looking at like the nucleus and the electrons. So how I like to kind of describe these is if we were to take kind of one molecule, you're going to have that electron cloud kind of floating around, and it's going to fluctuate in its dipole. As soon as you bring a second molecule in and getting closer, we're going to get that kind of same induced dipole effect. So you might get the electrons on one kind of moving to the other side, and then the electrons in the second one are going to be attracted to that positive nucleus. So these are going to attract in this induced dipole. So if we look at this line here, oh, do you see my pointer? If we look at this line here, we're coming in, we're getting closer and closer, we're getting to this attractive force as you're getting that induced dipole. But if you get too close and they come too close together, you're going to get repulsion of those positive nuclei. So these are very weak on their own, but they actually can contribute if you get a lot of van der Waals interactions to overall a very strong effect in proteins. So the additive effect of these can be significant. Okay, next, going from one of the weakest interactions to the strongest interactions, hydrophobic effect. So the hydrophobic effect is really going to help drive protein folding. So it's kind of like oil and water. When you put oil into water, it's all going to kind of clump and group together. So that's the same thing we really get with protein folding, is you get that kind of like collapse of this hydrophobic core. So one of the, I think, practice problems is, which law of thermodynamics is kind of driving this. Second law, perfect, thank you. So the second law of thermodynamics, and this involves entropy. So we're always moving towards that kind of positive entropy. So So how this works, great job guys, is we're getting like a water cage. So if you think about it, every single hydrophobic amino acid in a chain is going to have a cage of water molecules ordered and structured around it. So if you push these into one core to interact with each other, you're going to get the release of this kind of water cage. And then you're also going to get the more polar amino acids on the surface, and these are going to be kind of favorably interacting with the water instead of having them in this ordered kind of structure. So that's kind of how we're saying that the entropy is driving it, as you're getting the release of these ordered water molecules upon forming that hydrophobic core. So why do they form this cage? It's kind of the only way I guess they can like really cope with it. So they're trying to like shield it and protect it because the water really doesn't want to interact with it. Hydrophobic residues, they're greasy, they're not polar, it's not a happy interaction. So they kind of try and do their best to kind of fit around that in the most favorable way by kind of interacting with each other in this like cage. So that's why they really do want to push it together. So there's much more favorable interactions occurring. Okay. All right, amino acids. Love this part. All right, you guys are saying you have to know these. I'm sorry, I do. It's gonna help you, and it's really interesting. So you're increasing the entropy. So you're increasing the disorder. Therefore, you're increasing the entropy. Oh no, amino acids. The best part. Okay. General structure of the amino acids. We have our alpha carbon here, which is going to be forming our tetrahedral structure. So this is planar, but it is, remember, tetrahedral. We're going to have our hydrogen, our amino group, which is going to have a pK of about 9. So this is going to say that this is most likely protonated. And then we're going to have our carboxyl group, which has a pK of 2. So this is going to be found deprotonated. I should have had that deprotonated at our physiological pH. And then we're going to have our R group. So this is, someone said the R group. Yep. This is going to really dictate what properties this has. So is it hydrophobic? Is it aromatic? Is it hydrophilic? Is it polar? Is it charged? How's it going to be interacting? Someone said, how do we know the pKa? I'm going to give them to you. So we're going to go over that. So that's your basic structure, and this kind of tetrahedral structure is going to become important next week when we talk about folding. Okay. Boom. All right, you guys talked a bit about this in lecture, but and I think you got the hydrophobicity scales, and there's some students in the discussion board kind of asking about this. You know, how is it grouping? What's more hydrophobic? What is the, you know, asking about the characteristics. What I want you guys to do is take a second, look at the amino acids, and kind of think to yourself, how would you, if you had to organize this, how would you do it? Take into consideration, you know, the properties in them, and then just put it in the chat. Okay, you guys. Everyone's just doing the dashed up. That's it? That's all you guys got? Polar, non-charged? What if they're like bowls? Someone said branched. On a scale of which one is more painful to write? Cyclic. Sort by structure, how? Phenyl rings. Okay, if they look pretty, I think they all look pretty. Negative and positively charged. Acidic, basic, salt bridges. Ionic, chiral. Oh, okay. Aromatic, length. This is fun. Alphabetically. Functional. Oh, I like that functionality. That's cool. No one has said a blend yet. Interesting. A blend. Someone just did. What about amphipathic? No one said amphipathic yet. Whether you're long or not. Yeah. Okay, so as you guys can see from the chat, I know a lot of you guys just did arrows for one students. there's a lot of different ways we can organize these, and there's no kind of like correct organization. So some of the ones, some people do it based, I think someone said rings, so some people do separate the aromatic residues. Some people have a ambipathic, so some of these are kind of polar, but also they have some non-polar characteristics too, whether they're like protein shake or not. So there's a lot of different ways we can do this, and there's no like hard settings. And that's kind of what I want you to appreciate is there's no real kind of cut and dry lines. There are for some, but some of them can fit in bowls. Okay. I really said it was in my protein shake. That's hilarious. All right. So I did put some headings on these. So I did put hydrophobic. So these are, for the most part, hydrophobic. Even though some people say phenylalanine is aromatic. Oh, sorry, you guys don't hear me. Talk a little bit louder. All right, so we have alanine. So this is one of our kind of most basic amino acids, and sorry, not basic in terms of acidity, but basic in terms of there's not a whole lot going on. So we have just one methyl group for our group. So this is why alanine is commonly used in mutagenesis, so immunogenesis scan, because it's just not really intrusive. It's kind of this methyl group's more of like a placeholder. So you still have the tetrahedral structure, but it's not really contributing a whole lot to any sort of property. It's just kind of there. So if you want to see if the functional group of an amino acid is important, alanine is just kind of a good placeholder to see, you know, was that our group really necessary for structure or function. Phenylalanine, this has our large aromatic ring, so obviously this is extremely hydrophobic. It can also be found in stabilizing interactions. You can get pi-pi stacking to help stabilize protein structure through this aromatic ring. We have proline, and this is an interesting amino acid because we have the carbon chain that kind of comes back and links to its amino group. So this is going to have, sorry, I'm just looking at the group chat. The stars are all that. So this is going to have a huge effect on that structure. So remember, we have that tetrahedral character from our carbon, but this is going to kind of have this planar structure because we have this ring forming. So this is going to make it really rigid, and this is going to have a lot of consequences when we get to protein structures in terms of that flexibility about that alpha carbon bond. Okay, so it's going to make it quite rigid, and we're going to get into some of those properties when we get into secondary structure. But it's also fairly hydrophobic as we have that aliphatic chain. All right, more hydrophobic. We have our valine, isoleucine, and leucine. These are extremely hydrophobic. They're very greasy. They're often found in membrane proteins or in the hydrophobic core. Isn't isoleucine very nonpolar and hydrophobic? Yes, it is. Mutagenesis. Sorry, mutagenesis is if you were to take a protein, and let's say we had a valine, and we wanted to see if that valine was important, you'd switch it out with an alanine genetically. So you can like express a mutant protein where you switch those amino acids. So valine is going to be our beta branched. So I just want to take a second to go over branching. So our alpha carbon is going to be our alpha. So it's kind of like counting. So alpha, beta, gamma, and so on. So we have our alpha, beta, and then we have two gammas. Isoleucine is probably a little bit easier. So we have our alpha, beta, gamma, and then I can't count any higher in Greek, so that's about it. And then our leucine, we actually have a gamma branch. So what the beta branch means is how many kind of different substituents are on that carbon. So if we were to look at the beta carbon on valine, we have 3 R groups, so 3 different substituents, whereas if we look on leucine, we only have 2. So we have 2 hydrogens versus 1 hydrogen. So we have the same thing with isoleucine. We have 3 carbons and 1 hydrogen, so you're having 3 different substituents onto that carbon. So that's what it means by being beta branched, is like you have like an R group instead of that hydrogen. In terms of participation, if you guys are asking, take a look at the time for your first poll, and then you need an hour from there to your last poll to make sure you get your participation. Okay. Is branching increasing hydrophobicity? You're still going to have the same hydrophobicity if it's in a linear chain versus branched, but what else might the branching do? Yeah, so your beta branch is going to be 3 different R groups, 3 different substituents, and 1 hydrogen. Kind of weird how leucine is isobutyl. Instability, no. Interlock with each other, kind of. Decrease van der Waals, yeah, we're kind of getting there. Bulkier, favoring structures like beta sheets over alpha helices, yeah, perfect. Yeah, so you guys are definitely getting there. If we're looking at kind of the rotation about the alpha carbon bond, it's going to be more flexible in a leucine than in a valine or an isoleucine. So you're getting more bulk closer to that alpha carbon. So this is going to have effects on folding. And I think I saw some students saying, you're favoring a beta sheet over an alpha helix, which is true because it's easier to adopt that beta sheet conformation with that extra bulk than the more twisted alpha helical structure. So yeah, definitely has an effect on kind of how that amino acid likes to behave and be found. Okay, do the substituents need to be carbon? No, and we're going to see that in a second. Okay, so that's the beta branch. I think is everyone okay with that? How to identify a beta branch? Okay, great. Love the thumbs up. Okay, so we have our alpha carbon, and then if we go out in our linear chain, we're going to have our alpha carbon, our beta carbon, our gamma carbon, and then I don't know what it's after that. Oh, sorry, someone also asked about the gamma branch earlier. So this is our gamma carbon, and again, this has 3 different constituents or 3 different R groups, so that's going to be a branch. So it's not just linear. Then delta and epsilon. Oh, you guys are better at counting than I am. Okay, delta, that's it. Okay, perfect, thanks guys. Sorry, substituent, yeah, so that's anything, or R group is anything but a hydrogen. Yeah. Isoleucine, nothing group, would you count as a gamma carbon? Alpha, beta, gamma. So yeah, these would both be gamma. I think they usually have like a gamma and a gamma prime if there's 2. Leucine has a gamma branch, yeah. The beta branch is more important because this is what's adding bulk, and this kind of has, it gives more kind of properties. Like this branch is farther away, and there's larger, bulkier kind of amino acids like phenylalanine, tyrosine. So those qualities aren't really going to affect folding in that case. So it's more the fact that this beta branch is adding bulk closer to that alpha carbon that's going to be kind of favoring that beta sheet structure or kind of, adding less flexibility to those bonds. Isn't valine branched too? Valine is branched. It's beta branched. With beta carbon. So the branch is on which carbon? So we got branching on the beta carbon here. We have branching on also the beta carbon in isoleucine. And then in leucine, we have a beta carbon that's still linear, and then our gamma carbon also has a branch. Valine has 3 different substituents: carbon, carbon, carbon. Yeah, cool. Yes. Someone's saying the branch is farther away on leucine, so that's why it's not beta. If the branching has to be on the beta carbon, or if some people, I think, numbered it different. So if you want to number it like 1, 2, 3, 4, you start with the carboxyl, so it's 1, 2, 3, 4, to not over confuse everyone. But the branch, where are you describing the branches on which carbon that branch is occurring on? That helps make sense. Oh, sorry, it doesn't have to have, yeah, sorry, the substituents can be the same, sorry. They can all 3 of them can be carbon, but as long as they're just 3 R groups or 3 substituents, not hydrogen. Sorry, guys. I think that's, okay. There we go. Yes, they can all be carbon, they just can't be hydrogen. Beauty. Okay, I'm glad we got there. All right. All right, in terms of time, I'm going to get through the amino acids, and I'm going to give you guys a quick 5-minute break, and then we'll get going. Just because I know that this is a lot. 2 hours. Okay, next we have, I didn't categorize these. So they can fit into several different categories. So we have glycine. Some people say it's polar, some people say it's non-polar. It's kind of just nothing. It's the only amino acid that's not chiral. It's very flexible. 2 H bonds, perfect. That's why. So we don't have those 4 different substituents. It's less flexible. Yes. It's actually not less flexible, or oh, I think maybe you're responding to something else. Glycine, so we have that R group that's a proton or hydrogen, so it actually is a very flexible amino acid. So this is going to kind of favor some other structures, so like not necessarily always it can fit into alpha helices, but it prefers beta sheets because it's almost like too flexible. We're going to see it a lot in turns because of this flexibility. So this offers a lot of unique properties, and this is also why it's not usually used in mutagenesis, because it's giving more flexibility to that backbone than alanine might. Okay, tyrosine. So tyrosine is going to have the large aromatic ring as well as this hydroxyl group. So this is what I mean by having kind of 2 properties. So we have very, very hydrophobic ring, but then we have this polar group. So that's why it can be, it's considered sometimes amphipathic, because it has both of these qualities. Chloe, oh my god, what did I do? Okay. Okay, so it has a pK of 10, so this group can be deprotonated, but it has to be pretty basic environment to get that deprotonation occurring. We have our methionine, so we're going to have this sulfur group. So the interesting thing about this is it cannot form a disulfide bond because we have this methyl group. So in our cysteine, you can get that deprotonation, so you can get that exposure of the sulfur to form that disulfide, but you can't do that in the methionine. And tryptophan, again, has this large aromatic group, and it's really important in terms of absorption, which we're going to get into later. You can start quantifying proteins with this. You can do a lot of really interesting stuff looking at the fluorescence of tryptophan. Tryptophan. Yeah, no, actually, I think that is how some people do it, or kind of memorize the single letter codes. Tyrosine, yeah, methionine. Why can't methionine form a disulfide bond? Because you can't ionize the methyl group. You can't deprotonate that. So, and you need to in order to form that bond. Okay, moving along. Cysteine, so we have, again, we're taking that sulfur, we have a proton, so you can ionize that and expose the cysteine in order to form a disulfide. So it has a pKa of about 8. So this can be ionized. And then we have serine and threonine. So again, going over our beta branch structures, our serine has our 2 hydrogens. So even though this substituent or our group is on the beta carbon, we still have our 2 hydrogens. So it's not beta branched, it's still kind of considered linear. Whereas if you take our threonine, we have that methyl group. So we have the different substituents, therefore it is beta branched. Serine and threonine can form H bonds, which is really important for stability. However, they cannot be ionized. So this has to do with the stability of the negatively charged oxygen upon deprotonation, which is really unfavorable because there's nothing there to kind of help stabilize that. So that's why these groups don't have a pKa, unless I think theoretically they calculated them to be like really high, like you need a super basic environment. But for the most part, these are going to be protonated. I think they should be available like pretty soon, right after. Whenever I'm recording it, whenever they get posted by Dr. Patterson. We next have our kind of polar hydrophilic amino acids. Can you please repeat what you just said? About tutorials? They should be posted at some point today. We have our spargine and our glutamine. Oh no, slide. I'm just going to go back real quick. These groups do not have a pKa, is what I was trying to say. So they're polar, they can participate in hydrogen bonds, but they cannot be deprotonated. And that's due to the stability. Threonine has a pKa of 13 from the slides. Is it 13? I thought it was like higher than that. But that's kind of like, I guess, like technically you do need a really high pH in order to pull that proton off. But in terms of, I guess our arginine is 12.5. But we're never going to really kind of be in conditions with that kind of pH where we would see that deprotonation. Yeah, so if you have a super basic environment, so if it's pKa 13, you need to get to like pH 14 to really deprotonate those. We're really not going to see. So it can be done. It just doesn't really happen ever in nature. Okay, we have asparagine and glutamine. So we have this amide group at the end. So they're polar, but they're uncharged. These are good mimics of aspartic acid and glutamic acid, and this can kind of tell us if we were to like kind of do, let's say, immunogenesis, and you were to switch out asparagine with aspartic acid, is that charge important, or kind of the structure that's important? So if you're wearing roots in Starbucks, you deprotonate. Okay. We're getting into our acidic amino acids. So we have our aspartic acid, which is our D. So we have that carboxylic group, and our glutamic acid is going to have one more CH2 group. So it's going to be a little bit longer. And these are going to both have low pKas. So we're going to have a pKa of about 4, which means that this is a really good acid. So it's going to be found deprotonated most of the time or all the time at physiological pH. So it's a good acid. It's going to want to donate its proton. Our basic amino acids, we have our lysine, so we have our amine group. So its pKa is 10.5, so this is going to be mostly in the protonated state. Our arginine, again basic, it has this guanidinium group with 12.5 pKa, so it's going to be protonated. And then histidine, we also have this imidazole ring, and it has a side chain of about 6. Its pKa, so it's going to be found at physiological pH deprotonated, but because it's actually close, I think you guys talked a bit about this in lecture, to physiological pH, sometimes when it's found in active sites, can we get a bowl, please? I think we're going to be getting one soon. We're learning about amino acids. Give me a second. So histidine is going to be found in that active site. And depending on the environment, you can kind of sometimes shift that pKa, kind of like we see with our hydroxyl groups, where depending on what they're attached to, they're given more stability in order to become deprotonated. And the same also happens with histidine. So depending on the stability that is found in the active site of a protein or enzyme, it can kind of shift. So this can sometimes be found protonated at physiological pH due to the shift, and it's super important and found in a lot of active sites for a lot of enzymatic reactions because of this really unique property. Okay. So someone asked, how do we know? So this is just a summary of all the ionizable amino acids and their kind of relative pKa. Okay. I'm going to give you guys a break, and I'll give you guys 5 minutes or so, and I will put up a poll for those of you that want to head out. There we go. Does the poll of the bachelor count? Yes, the bachelor counts. I will try and do that. I'll put up a question at the beginning so it could start your time. Okay, I'm going to try to go back through. Get disconnected in the middle, come back again. I'm not sure about the disconnection. You could probably check with Dr. Patterson if you get cut off. The proper drawing of the amino acids kind of depends. I think people draw it differently. So they're all the same structures. It's just kind of how best you can, I guess, visualize it. So there's no, like, I guess, correct way. Some people are going to draw it slightly different in terms of, like, orientation. Is there any other biochem texts you could recommend? Like books? Sorry, Stephanie, I guess, is asking, like, you want, like, textbooks? Or, like web pages? What was the last basic amino acid? Lysine, arginine, and histidine. Yes. Can you go back one slide? Yes, I can. What if we missed the bachelor question? So it's going to count from whatever the next poll question you answered. Do you have to answer all the poll questions? No, so you just kind of have to answer, I guess, a couple within the hour span or over an hour to show that you were here. You have to memorize pKa's. You should know the pKa so that, like, if you are given an amino acid chain, you can kind of identify, like, the overall charge of that polypeptide chain. So it'll be helpful, especially when we get into kinetics later, in looking at active sites and binding pockets and which amino acids are found in those binding pockets. It'll help if you're like, oh, that's a charged residue that's going to interact favorably with, you know, the opposite charge. Are they basic because of the charge? Yeah, so they're positive, so they favor the protonated kind of positive state. I missed the first question, so I mean, I can't leave. Yeah, you guys should just try and time it, or just stay for the whole thing, and then you don't have to worry about that. But I understand you guys have a lot going on. So yeah, maybe set yourself a timer from the first poll you ask. Resources. I guess from resources, the chem, I think it's like WikiChem I find is like pretty good. I don't know, I don't use like a specific thing. I guess I always had like big textbooks, so I always relied heavily on textbooks as a student. But there's, I guess, I don't know, so I don't have a specific like web page, but I can look into that for you. Physiological pH is above the pK of a group. The group will be deeper. Oh my God, there's 85 messages. Ah. Okay. Yeah, so if your pH is above your pKa, you're going to be deprotonated. Who's Chris? Someone says, why memorize anything? It's just good to know. But yeah, when you're doing your assignments, you can obviously definitely have a sheet beside you to refer to, or your quizzes. Clearly they like some beef. Like we prefer chicken and beef? Oh my gosh. Okay. You guys are chatty, I can't even catch up. Okay. Who's Chris? Oh my gosh, this is hilarious. Some resources. Okay, I will try. I will try and get some resources for you guys for the next one. I'll write that down. Okay, we're gonna get back into it. Okay. Sorry, there's a lot of comments here. I'm trying to them all. Okay. All right. Okay, we're getting into practice problems. Okay. Here's an example of 4 lysines. What pH environment or what pH range, given your knowledge of the pKa's for lysine, would these be found in? Look at the protonated state of each functional group. Ooh, someone's on the ball. Okay, less than 2. Okay, someone said less than 2, 2 to 9, greater than 9, greater than 10. Close, very close. First 2 are good. So if we were to take a look at the first one, what is something we notice about all the functional groups? They're all protonated. Perfect. So what pKa do they have to be below? 2. Awesome. So in order for them to all be protonated, we have to be below the lowest functional group, which is 2. Love that. Okay, charge plus 2. Next one. Carboxyl's deprotonated. And then we have our protonated amine and our protonated R group. So now what do we have to be? Two to 9. Yes, perfect. So we deprotonated it. So we have to be above that. But we have to be below 9, otherwise our amine would be deprotonated. Perfect. Our next one, we have our carboxyl and our amine deprotonated, but our R group's protonated. Perfect, so we're between those, so we're over our pKa for our aiming group, so 9, but we still have to be below the pKa for our R group because that's going to be still protonated. And then our last one, we're all deprotonated. Beauty, above 10.5. Great, so how you go about that is, again, look at deprotonated rates, therefore, what do you have to be above and below? Where did we get the values from? If you look at the previous chart, I gave you just kind of a general range. There's more exact numbers if you guys have to calculate the concentration. So like if you're using the Henderson-Hasselbalch equation, like definitely go into the decimals. But this is just kind of like a general pKa for the side chains. Okay, draw the various ionized forms of glutamine and indicate which one predominates at pH 1, 7, and 11. So also what type of ion, I guess, is that the right word for it? Or are we going to have based on the charge? So our pH 1, what are we going to see for all the first ones? Both are going to be protonated. Yeah, so what charge on this going to be? +1. So what is that? Positive cation. Yeah, there we go. Perfect. So we have a cation. pH 7, what are we going to have? What's our charge? Neutral, yeah. And so if we have neutral, but we have 2 charged species, what does that consider ion? Love it. Great. Okay, so Zwitter ion is when you're neutral, but you have a charged species. And so we're going to have our protonated amine group is going to be charged, and our deprotonated carboxyl group is going to be have a negative charge on it. And then lastly, at pH 11, negative anion, beauty. So we're now above all the pKa's. Everything's going to be deprotonated, therefore we're going to have a negative charge. Excellent. Practice problem. Salt bridge can form between what and what? Sorry, I'm going to go to the poll. So this is where it's good to know your side chains by saying if you get the just the name, you should be able to be able to do your action. Be making a poll. It takes a little while. 8. Okay, there we go. Poll. Can I go back to the last slide? There we go. Is that okay? There we go. Okay. I'm going to put the question back up so the students can see. Yeah, so this is why I know you guys definitely, I know it's early in the course, so you're not going to have these all memorized yet, but this is just kind of an example for what you guys should know going forward. Okay, majority of people are saying C. Excellent. So I finally know one of the amino acids. You guys are killing me here. All right. So we have lysine and aspartic acid. So we have our basic amino acid and our acidic amino acid. We have that positive charge and a negative charge. Those are going to be interacting through an electrostatic interaction, forming that salt bridge. Okay. Let's see. Okay, I'm going to put up another poll. Under what circumstances would a hydrogen bond form in a protein? Give me a sec, putting up a poll. A, B, C, D. Here we go. Yeah, sorry, I do use aspartic acid and aspartate interchangeably. You can use either one. I can switch it up. I don't know why. Ooh, we're getting quite the spread here. This is interesting, guys. Okay, love this. Okay, we are for the most part, I think most of us are guessing. Okay. For the most part, everyone's saying B, which is the correct answer. But we are getting quite the spread. So I'm going to go through these. Okay. A deprotonated cysteine and a deprotonated tyrosine. Do we have a hydrogen bond donor in this case? No. Perfect. So there's no group donating that hydrogen bond. So even though cysteine and tyrosine are able to form hydrogen bonds, you need that hydrogen. Let's go to B. A glutamate side chain is exposed on the surface of a soluble protein. So if it's on the surface of a protein, and we have a glutamate, it's probably deprotonated. How could we form a hydrogen bond? Or what would we be forming a hydrogen bond with? With water. Perfect. Yeah. So remember, water is going to help stabilizing protein structure, and it's like super important. So that's why I want to kind of highlight you can be stabilizing it with, in the environment. So it is kind of participating in those hydrogen bonds on the surface with water. Okay, C, an asparagine and serine are located on different faces of an alpha helix at physiological pH. So could an asparagine and a serine form an H bond? So, yeah, but they're on different faces. They're too far apart. Perfect. So yeah, that's what I was trying to highlight here is that even though they have to be in close enough proximity and the right orientation in order to form those bonds. And then the amine bond and a peptide backbone is accessible to an aqueous environment. What's wrong with this? What's wrong with this statement? It's an amide bond. Yes, functional groups. So amine, it's an amide bond in the peptide backbone, so that was wrong with this statement. If it was the, if I said, the amide bond, the backbone can definitely interact with water to form those hydrogen bonds. Asparagine was actually named after asparagus. Okay, answer is B. Again, okay, here we go. Another poll. I'm gonna make a poll and then is there an E? Yeah, there's an E update. Okay, here we go. Okay, some students are asking about C and D. So it was an amide bond, not amine, and the amino acids have to be in close enough proximity in order to actually form a hydrogen bond. Thanks, guys, in the chat. Okay, you guys are doing well on this one for the most part. We got E. So the hydrophobic amino acids have side chains that are nonpolar and uncharged. So we want those aliphatic chains. So C is possible if they're in close proximity. Yeah, for the past question. If they were close enough together, they could. All right, so good job, guys, on this one. E, lovely. All right, another one. So much participation. Here we go. Update. Which amino acid is not chiral? The answer was E on the last one. Okay, perfect. You guys are doing well. Glycine. So remember, there's only one non-chiral amino acid. So you need 4 different substituents in order to be chiral. Glycine has 2 hydrogens, so therefore it is not chiral. Ooh, sorry, just to all go this guy. There we go. All right. Okay. This one's going to be, I'm not going to need a pull for this one. What would the titration curve look like for glycine versus arginine? Why? So how many, I guess, let's say inflection points are we going to get? And what are causing those inflection points in glycine and arginine? Two versus 3. Titration creation equals heartbreak. You don't need to know them in like super depth, but you should like appreciate like what the inflection points mean. So for the most part, you guys are getting it 2 versus 3. So it's going to be due to the R group pKa. Yeah. So the different points of deprotonation. Perfect. Well done, guys. So for each pKa, you guys are talking about like that buffering region. We're going to get buffering within kind of 1 step of the pH. So, but you're going to get an inflection point for each pKa, so upon each kind of deprotonation event. So glycine, we're going to have our carboxylic acid as well as our amine, both kind of undergoing those deprotonations. Where in arginine, you're going to have 3 because you have your 2 termini plus your R group. So you're going to get those 3 inflection points. And they're going to occur like at the pH when your pKa equals your pH. So, yeah, you don't have to, I guess I don't know if you need to know these super in depth, but like just kind of appreciate what that the pKa's are responsible for those inflection points when you're getting that buffering around your pKa. Okay, this one's going to be fun. Calculate the approximate PI for the following peptide. Why does arginine have 3? Arginine has 3 because it has 3 different pKa's. So those are 2 terminus plus 1 side chain. Thank you. Yeah. Yes, confusion. Okay. Really, I forgot what PI was. Oh my god, you guys are on fire this morning. I love it. Okay, PI. So this is when you're going to have your neutral species. So at what pH are you going to have a neutral polypeptide chain? Flavor. Isn't PI for 1 amino acid? Nope, you can have it for a whole polypeptide. So we're gonna go through this, but I want to give you guys a second to try and work your way through. So okay. Whole polypeptide charge 0. Yes. So what you're gonna want to do is make everything protonated. What is the charge? You then have to increase the pH and slowly kind of deprotonate groups until you hit an overall charge of 0. And then you're going to average the 2 pKa's, 0 falls somewhere between. So you're averaging only the 2 pKa's that surround that neutral species. Let me know what you guys get. I have it all written out and I'll show you guys. I just want to see if anyone. Okay, some people are getting close. Yeah, 7.1. Okay, yeah. Confusion. Oh no, guys. Okay, 7. All right. 7.6. You guys are all pretty close. Mercy. Okay, let's just go through this. All right. Okay. If we were, let's try this. pH 1, what is the overall charge of this polypeptide chain? So this is where I was saying you need to be able to see the 3-letter code or the 1-letter code and think about its structure and be like, oh, this has a pKa. Okay, so +2. Remember the termini. +3, yes. Okay, here we go. So, how I find is the best way to calculate the PI is I like to put this in the fully protonated state and then work my way up. So let's set it to pH 1. So at pH 1, you're going to have your protonated amino terminus. So this is going to be a +1 charge. Our cysteine is going to have, it's going to be protonated. It's going to be neutral. Our arginine is going to be protonated. This is going to be positive. Because remember, we have that guanidinium group, which is going to have the protonated nitrogen. Phenylalanine doesn't have pKa. Histidine, it does have a pKa. It's going to be protonated. Again, nitrogen, when they're protonated, has a positive charge. Aspartic acid does have a pKa. It's going to be protonated, but it's neutral. Glutamine, no pKa. Our free carboxyl is going to have a protonated, so it's going to be neutral. Someone says, what is the free amino? So this is just your terminus. So your NNC termini for all your amino acids, they're going to be like tied up in the peptide bond. So you don't need to look at them for kind of like each amino acid in the chain because they're not going to be contributing to that ionization. But your termini are going to be free and exposed. So those are going to undergo ionization. So what I like to do is I list the pKi values of all the amino acids from most acidic to basic. So where do I get these values? I think this was posted in class. So someone says, can you upload these slides later? The tutorials are going to be recorded, so you can go back and look at these and have them. So yeah, so someone says you just find the charge where it's neutral. Yeah. So I list them from most acidic to basic, and the idea is that it's most, if pH equals 1, we're going to have a charge of +3. So how many species do we have to deprotonate in order to get to that neutral charge? So we're going to look at this stepwise. So if we take our free carboxyl, and we're going to, that has a pK of 2.2. Let's say we increase our pH. We're now going above that. So we're deprotonating that group. We're now having a -1 charge. So we're lowering the overall charge to +2. Now above the pK of 2.2. Our next pKa is going to be our aspartic acid at around 3.9. So if we increase our pH above 3.9, we're deprotonating that carboxylic group. It's now becoming negative, and our overall charge is going to be +1. Our next group is going to be our histidine. So again, that's at 6. If we increase our pH above pH 6, we're deprotonating that histidine. This was originally a +1 charge. Upon deprotonation, you are now making it neutral. So now we have 0. So we have our neutral species. Now the thing is, what's the next pKa? It's going to be our cysteine R group. So if we go above 8.2 now, we're deprotonating that cysteine. We now have a negative charge, and we're going to have a -1. So the idea is now we're going to want to average these two pKa's, and our PI is going to be between them. So we take our histidine PI, which is 6, and our cysteine PI, which is 8.2, average those, and we're going to get 7.1. How did I do? Okay, I'm going to go look at this chat. Why do you increase the pH? So you're looking for the pH at which your species is neutral. So that's why I find it easy to kind of put it all positively charged. So if you have a +3 positive charge at pH 1, then you're going to need to deprotonate 3 different pKa's in order to make that neutral, and then you can average the pKa's around that neutral species. Someone said, yeah, so it's knowing which structures, charges, and determining, yeah. Someone said, do we need to memorize the pKa's? If you're asked a question like this, like if they want this specific, then you should be given a chart. Like if this was an exam, you would have a chart with these pKa's. For your quiz, I'm assuming she'll probably give you the pKa's of what you need to know if you get a question like this. But if not, like you have this in, I think, your lecture slides, so you can refer to that. Does it have to be the middle acid or the acid on the chain is fine? I'm not sure what you mean by that. Why do you use histidine and cysteine? It's because between these two kind of pKa's, you have a neutral species. Oh, someone says, why does ASP have 0.2? Sorry, so these charges here that I've kind of highlighted are the overall charge. So if I go back, fully protonated, this is the charge of each amino acid to give us the total charge. And then I'm saying here, the total charge is +3 upon deprotonation of your free carboxylic acid, you're reducing the overall charge to +2. Some students are asking about needing to take chem. It's a bit chem heavy in the beginning, just to kind of understand the ionization and stuff like that. But it's not like you don't need to take another course. Just maybe review like these kind of ionizations a bit. You'll be fine. You got this. This means the whole polypeptide is neutral. Then you do it again. So yeah, even though you're going to have some charged species, the overall charge is neutral. And that's what we're looking for. Is there going to be time at the end to ask questions? Hopefully. The ionization of the amine and carboxylic. Someone says, how come the ionization of the amine and carboxylic acid are taken into account? So they're tied up in the peptide bond. So they're no longer going to be able to be ionized. So it's just the terminal amine and carboxyl group that we need to take into consideration. What is happening in step 3? In step 3, we're increasing the pH above each pKa and slowly deprotonating them. Okay, how would I say this one more time? And then hopefully we'll see how it goes, and then we'll get to some of the other questions. Okay, just to go over it one more time. We're going to, the way I find it's easiest to find the PI is I start at pH 1, fully protonate everything, look for the total charge. So we're going to have a +3 charge on the peptide at pH 1. So you can think to yourself, you have to deprotonate 3 functional groups in order to get to a neutral charge. So how I do that is I kind of list the pKa values from most acidic to most basic to see, okay, how much do I have to increase the pH to deprotonate those 3 groups. So we're going back to this. So this is the overall charge of pH, let's say 1 here. It's going to be +3. And if we cross the pKa of 2.2, say our pH now equals 2.2, we're deprotonating that free carboxyl. So we're going to have a -1 charge now on the carboxyl. So we're reducing the overall charge to +2. If we now go to a pH of 3.9, we're deprotonating the aspartic acid. So now we have another negative charge. So we're decreasing that overall charge to +1. And then again, now if we go to pH 6, we're deprotonating the histidine, so we no longer have a positive charge. We're getting a pH or a overall charge of 0. So we've hit that neutral species. So if we cross the next pKa, 8.2, we're going to get a -1 charge. So now we can say, okay, we can average these two groups to say between histidine and between cysteine, so between 6 and 8.2, we're going to get a neutral species, and then we could average those. So basically, we're finding the medium of the pKa. Yeah, when you get to that pH of 0, you want to look at the medium of that. How do you know whether deprotonate or protonate based on the pH? So if it's gonna be deprotonated if it's above the pKa, or if it's going to be protonated if you're below the pKa. So you need to know the pKa's of these amino acids. It says you have to deprotonate 3. Yeah, so you're deprotonating 3 with the lowest pKa's. There's definitely, I think some students are saying other ways to calculate this. There's other, like, this is the way I find the easiest, but if there's a different way that you guys want to do this, this is not like you have to do it this way. Whichever way kind of makes the most sense to you, definitely do. Someone's asking about the free carboxyl. So we have the carboxylic group. The carboxylic acid on the C-terminus is exposed on the glutamine. Okay. Can the PI be between anywhere in 6 and 8.2? Just easier to find the average because remember you're going to be kind of half deprotonated, half protonated. It's like 6, so that's why whatever's in the middle. Okay, I'm going to move along just so we can get through these, and then hopefully we might have some time and I can stick around a little bit after to go over this if anyone has any more questions. Okay, so we're going to practice problem, given an amino acid of the non-ionizable side chain, what percent of each termini would be protonated if the pH equals 7? So it's kind of, you'll see sometimes in textbooks, the backbone drawn like this, where you have this deprotonated amine group, but a protonated carboxylic group, which doesn't quite make sense if we think about their pKa's, when they really should be ionized. So using the Henderson-Hasselbalch equation, we can actually see what percent of these are actually going to be found. So what percent of each terminite would be protonated? So do the Henderson-Hasselbalch equation for the amine group and the carboxylic acid group, and I just put in 2, oh I did 8, oops, 2 and 8, you can probably do 9, 2 and 9 for these groups for the pKa, and then kind of calculate the ratio of those protonated versus deprotonated form at pH 7. Let me know what you've got. I'm just going to go back and look at 9 and 2, yeah. I did use 8 in this example. I'm sorry, guys, I didn't update that. That's my bad. Again, I just kind of generalized the ones. So if we look at our groups, our Henderson-Hasselbalch equation, let's look at our carboxylic group. pH equals 7, our pK is about 2. If we were to look at the ratio of our deprotonated versus protonated, we have, what is that, 100,000 times? 10 to the 5. So this is saying the majority is going to be in the deprotonated form. So this is just an equation I found to find fractions. There's multiple ways you can do this. So our actually protonated species is going to be like 0.1 or 0.001%. So it's at physiological pH, you're going to have almost basically all of it is going to be deprotonated. Whereas if we look at the amino group, I think, as I said, this should have been a 9, sorry. If we look at our ratio, we're going to have 0.1. So this is about 90%, probably more if I had the right number, is going to be in the protonated form. So this is kind of just showing that the correct way to represent the backbone is this example here, where we have that zwitterionic species. And this is an example where using the Henderson-Hasselbalch equation that you can actually calculate exact concentrations. Someone's saying, what is the FAH? I actually just looked up how to convert fractions to percentage, and this is what they gave me. But there's other ways you could do it. Sorry, what is supposed to be a 9? I think the average for the amino terminus is 9 instead of 8. We were using 9 before, so I'm sorry I forgot to update that. So this is, where did this come from? So you guys should know the approximate pKa of the termini. I don't, this type of question, you might get asked to use the Henderson-Hasselbalch equation in a quiz, but the fraction is just kind of showing you guys the populations that exist of these different species. But if you get a question, you'll probably be asked for the exact concentration. Okay. Review question, aspirin is weak acid at pK 3.5. Absorption requires passage across the plasma membrane. The rate of absorption is determined by polarity of the molecule. So you need a more neutral species in order to cross the plasma membrane. So if the stomach has a pH of 1.5 and the small intestine has a pH of 6, where's aspirin going to be most likely to cross the plasma membrane? Someone said the stomach. So why in the stomach? Did I mess that up? Oh, someone. It should have been plus log. Sorry about that. You're gonna have to update that question. Perfect. So it's protonated. Yes. So if we're below the pKa, so we're gonna have a protonated species. If it's therefore, if we look up oxygen or aspirin, protonated species is going to be neutral. Therefore, it's going to be more readily absorbed in the stomach. Okay. Next question, we're going to go into cofactors. I think there's been a lot of stuff on the board about this. There's two different kinds of cofactors. Cofactor is kind of an umbrella term. So we have our essential ions, which are inorganic, and then we're going to have our organic. So we have essential ions, inorganic, coenzymes, organic. Both of these groups can either be loosely bound or tightly bound. So if they're tightly bound, they're going to be really embedded in that structure, and they can either be bound covalently or non-covalently. Whereas the loosely bound ones are going to kind of come and go in contact with the protein, but they're all really essential for structure or function. Okay. All right. So we have our example is our heme group, which is going to help transport iron, and this is going to be very tightly bound, which is going to be a prosthetic group. So this is just kind of like the different layers of cofactors, whereas cofactor is the umbrella term for all of these. So to kind of look at, describe two important roles of protein cofactors, and just in the interest of time, I'll give this to you guys. So these are really important for structure. I think there was a sugar binding protein Dr. Patterson used, so upon calcium binding, it kind of opened up this structure and allowed it to interact with its substrates, or it can actually assist in catalysis or some other function. So sometimes these are needed to participate in like cleavage or binding, or in heme, it can transport. So there's a lot of different, tons and tons of different things these cofactors can do, and it's all specific to the protein that they're in. So to show you guys these cofactors, we're going to learn about pyruvate dehydrogenase in metabolism. It has, I think, six cofactors that are involved, and we can categorize these and these different kind of terms. So whether it's an essential ion, coenzyme, tightly bound, loosely bound. It is completely normal that you've never seen these before. So what I'm going to do is I want to show you structures of these to kind of show you the difference between what it means to be tightly bound or loosely bound. So our TTPP, so these, just to kind of show you on the left-hand side here, this is our actual protein. So the pink is the protein and the green is the protein. And you can see we have this structure, TTP, embedded into this protein, as well as this magnesium iron here. It's coordinated by these amino side chains, like really strongly embedded in that structure. Or FADH, same thing. It's embedded in that protein structure, and these dotted lines are showing the stabilization. We also, our other one, we have lipoamine. So it's this large kind of group here in black attached to this E2. And we have our functional group that's attached on the end. And the interesting thing about this is it actually kind of binds this functional group and moves it to the different active sites within the protein itself. So you can see that it's kind of a part of this protein. Our coenzyme A, you can see here in the reaction, it kind of comes and goes. So it comes into contact and picks up its functional group, and then it's released. Our NAD+, again, it's going to kind of come in, undergo that oxidation reduction reaction, and then be released. Are all those examples of cofactors? They are. So given these, seeing them, so this is not something you guys have to like memorize, I just kind of want to show you the difference between what it means to be tightly bound and loosely bound. So these structures that are like embedded in that protein are going to be your tightly bound structures, whereas if you have coenzyme A and NADH, these are going to be coming into contact, undergoing whatever reaction they have to, and then leaving. So these are going to be our loosely bound. So then based on that, we can organize them. Magnesium is an essential ion, and you could see that that was embedded in the structure, so it's going to be tightly bound. And then our coenzymes, which are organic, we're going to have our co-substrate, which are loosely bound, so that NADH and our coenzyme A that kind of came in contact and then left. And then we had these other structures that were really embedded, so those are our prosthetic groups. Ligands in our project proteins are cofactors, right? I guess it depends for your PDB. So sometimes they crystallize the structure with the ligand just to give it stability, or they want to capture that interaction. So I'm going to be disconnected in 5 minutes. So sometimes they crystallize it with the structure just so that they like capture that or stabilize it. So you'd have to like kind of look up your protein and see, is that the ligand or is that the coder? Are all tightly bound cofactors prosthetic groups? All organic tightly bound cofactors are basotic groups. Yeah. Are these slides posted? Not with the answers, but the recording is going to be posted. So you can go back and look at the answers. Prostatic group is a tightly bound organic molecule. Can you teach this whole course? Oh. Thanks, guys. Sorry I didn't save much time questions. We'll hopefully make the tutorial next week a little bit shorter so that there's at least 10 minutes or 15 minutes for questions. So sorry about that, guys. No problem. If you have any questions, feel free to email me or post on the board. I think I have 5 minutes left if anyone does have any questions here. Oh, I think I'm going to stop recording. How do I do that?